Mexico，然后我们是台湾Proteintech的总代理。那我们今天邀请到是来自德国的 Dr.Klaus Herick。那他曾经在Proteintech 或者 ABreal 等等国际公司担任一些国际职务，像是一些产品管理、营销等等新产品推出。那他在2015年的时候是加入到了德国的纳米抗体的研发平台 Proteintech。那并在那边担任他们的行销以及销售的副总。那在2022年的时候 ，Proteintech 并购了 Proteintech。那之后，我们的 Klaus Herick 呢，他就来到Proteintech 的体系里面，那去为 Proteintech 系列的 antibody 的一系列的产品线。那他主要担任的职务呢，是在亚太区的商业总监的部分。那今天再来，他会为我们介绍就是关于 Proteintech Genomics 这一块新出的一个关于单细胞多体学相关的一个产品 multi-pro， 以及对于single cell sequencing 的应用这样子。主要我们都欢迎他。
Didn't understand everything. I heard my name. You introduced me as another person. So thank you very much for showing up for our workshop or our introduction of the multi-pro product, the oligonucleotide antibodies and antibody cocktails. I'm very pleased to present this very new product line for Proteintech. We had to fight with the technology, and somehow the text is a bit strange, and the font sizes have been mixed up all over, but the graphs are okay, so apologies. Before I go into the content of this presentation, I want to ask you, are you all familiar, are you using SciSec or single cell analysis technologies? I'm asking because if you know better than me, I don't explain that to you guys, but you may be able to explain something. I know I with SciSec and single cell technologies. Yeah, no. Okay. So I will explain. So, just a few words. So I will introduce our company, Proteintech Group, and I will introduce SciSec and the some idea of the workflow using this oligo-labeled antibodies cocktails, show experimental data that all of the presentation, and a few words on the product. Here in Taiwan, Proteintech exclusive like ABreal. So Proteintech Group, it's an American company based out of Chicago and Illinois. It started 22 years ago on antibodies in the United States. That's where we have our Proteintech brand, if you like. Next to the roller vessels, we have some cytokines. We call this human time. Then we have an antibody-based reagents, these are actually manufactured in Munich, acquired by Proteintech 2020, so almost 4 years ago now, and that's the way how I think Proteintech, and a little bit less than targets. Then it's polyclonal antibody, that is monoclonal antibody, that is recombinant antibodies. What do you antibodies? The answer is very simple. Every antibody that was specific enough that we could label and that work with our application. So this is actually SCRNAseq. I have to admit that this multi-omics phase in research, they love abbreviations. I don't know why, but they have introduced a lot of new abbreviation and what is it? Let's see. This indexing, so marking the transcriptome as well as the proteome. It has been introduced in 2017, and it means that we can detect single-cell RNA, so the transcriptome, as well as cell surface proteins by antibodies binding to cell surface proteins that are shown here. So intracellularly, no proteins are safe. So it is limited to cell surface proteins only. So now, for the first time, available is the SciSec solution for intracellular antibodies. So in our cocktails, we have a combination of extracellular antibodies targeting extracellular targets as well as cytosolic targets, nuclear targets, and that is for the first time commercially available, and it has been designed to work exclusively with the 10x Genomics single cell flex system. So for the experts, this is the format of the conjugation, or the conjugate we're using, or the oligo that we're using, PCR handle, the antibody barcode, the capture sequence. We don't use a unique molecular identifier because that's already part of the capture sequence. And it's a so-called 5C-flex format. And I like to point out again, it's exclusively for use with 10x genomic single cell gene expression flex products. And that means with and without multiplexing. Now, how does an experiment work? So cells are stained, then the cells are partitioned, and cell lysis is being conducted, and what we get is mRNA and oligo capture. That means the mRNA transcripts that are in those cells are binding to probes, the antibodies as well, the oligos of the antibodies as well, and they are captured cell by cell, converted into a sequencing library, sequenced, and then analyzed. Like here for the CD4 example, here we have the RNA expression of the transcriptome, the RNA count, and here we have the protein expression for those cells. I will talk more about these UMAP displays later on. Now, how do the data look like? And here, just as an appetizer, I will show this data again later. Secreted proteins, interleukin 8 transcription factors, that's NFKB beta 2, KB2, and here phosphorylated proteins, that is beta-catenin phosphorylated at position 675. And what you can see here on all of these UMAP presentation is first a grouping presentation of the cells according to the cell types. So here we have the alpha T cells, here we have macrophages, here we have natural killer cells here, and the blue are B cells. So that's kind of the grouping, the clustering of the cells. And then correspondingly, we have here the RNA count, always, and then the color is an indication. Here the color is an indication of the cell type, and here the color is an indication of the side of the expression. And yellow means low, and purple means high. And here on the left side, the protein data, so the antibody data. Also here it's yellow low and purple. You can see there is a lot of overlap of these data. Here for the NKB2 transcription factor, you again see a lot of overlap, but also differences. You can see here, there is a transcriptome here for those monocytes is on the medium level, on the expression level, it's very low. So the transcripts are lots of mRNA present in those cells, however, express protein not. And then here, for the beta-catenin, the phosphorylated version of the beta-catenin position serine 675, you won't bring these to displace the quantity of the RNA, the transcriptome, the RNA count, in line with the proteome data, the protein expression. Because the transcriptome does not directly control the phosphorylation of the transcribed proteins. Now you may ask yourself the question, this is a lot of overlap. Why should I do the experiment twice? Why should I spend extra money for extra reagents? And very simple for two reasons. First, the data quality is much better when you can show that your RNA data are going along with your protein data. You can be much more confident of the data. Second, there are differences. You just need to find those differences, and we as a developing company, we always have to show that our systems, our products are working. So we also have an interest in showing a lot of overlap so that we can trust our antibodies and our antibody cocktail. And finally, here there's a non-correlated information between the transcriptome and the proteome for the phosphorylated. I found our approach with the phosphorylated proteins most striking. Now, a few words to the experimental workflow. It's an X instrument, so you have to fix and fertilize to hybridize your probes. Then you prune those, partition them into gems, the pruning system, and you ligate and extend it in these so-called gems, these little bubbles that are formed, these emulsions. You construct libraries for the protein expression as well as for gene expression, do a lot of sequencing work, and then spend a lot of time with the data. This in order to get meaningful data. How does the workflow in the laboratory, if you like, look like? So you have your single cell suspension. We are just talking about staining. That's where we are focusing. So the 10x workflow with the chromium single cell gene expression flex stops, if you like, starts with the FC block. And now the multi-probe workflow kicks in with a rather short 15 minutes 4% PFA fixation. Then we parallelize and block the cells. And then we do the antibody training. As you can see, surface proteins, cytosolic proteins, low-level proteins. And then the 10x genomics workflow continues with 1 hour 4% PFA fixation. So what we are basically introducing is an additional fixation step here. that has only a minimal impact on the RNA performance. Looked into this. So here you can see you have a graph on the 10x fixation step only, and here the protein take 10x fixation step. But first, when you look into the cell count, you will see two very different. And, that is okay, because the same show up here and here. When you look into the total RNA count, you can also see the same color show up. That means the RNA count is also the same on the Proteintech method, the fixation step, whereas the 10x step. So it's our scientists say it's only a minimal impact on the data. They can't exclude there is an impact. We also have developed what we call an enhanced blocking reagent that we really urge everybody to use it. And here it's shown for the example of the key 17A. That's the standard ESA C block, and here it's the enhanced blocking reagent plus C block. And what you see first here on the cell type graph on the very right adjusted new cells show. You need to believe me that these cells are somewhere here. We believe that this PBMCs are from donors, healthy donors, that these may be either granulites that have survived don't survive sample preparation, or that these are other very seldom cell types actually that can be seen here, also given an indication that our blocking is more sensitive and providing lower backgrounds. So we highly recommend to use our blood. Okay, so I like to show experimental data starting with secreted proteins, and you may ask the question. We call it intracellular science, and Klaus starts with secreted proteins. It has a logical problem, I agree, but what we stand here on these PBMCs that we have stimulated with either LPS or PMA. I think it's 4 hours stimulation. With this time period, we have used commercially available protein transport inhibitors. So the expressed proteins, the secreted proteins, interleukin 1 beta, 17A, and interferon gamma are actually accumulated. So after that, to come up with an experiment without using the protein transportation blockers. So we will see how the data look like. The protein transportation isn't blocked. But now I'd like to draw your attention on interleukin 1 beta. Here you have the cell count. Those cells here is and those macrophages that are here in this area have rather high expression on the RNA level, high RNA count, but on the protein side, the protein is highly expressed. It's the lowest count that we have. So here we have a disconnect. So something is going on. Here for the interleukin A and these T cells, yes, maybe T cells, same goes to also for the So you always have you need some time to spot the difference. I acknowledge based on the resolution of this screen that time possible. Transcription factors also very interesting. So here we have TCF. To draw your attention to this part of the microphone. Here we have rather high protein expression according to the antibody data, and the RNA transcript database, we have only very, very low expression. Here for 2, we have found 2 cells that are very rarely found in PBMCs, and again, as you can see, the clustering of the cell is protein by protein or RNA construct by RNA construct. Phosphorylated proteins, here it's, I think, difficult to do the overlay. 2 examples are shown: phosphomethyl-glutamine, serine 600, 298, and it is still possible to really show good correlation of mRNA data, transcriptome data, protein data, because it's not directly the population is not directly correlated to the transcription process. So that's probably the most striking point of using intracellular science. Then hydrosonic proteins, here a few shown that also show some areas that are like here in this area of the cytotoxic T cells that you can see here, highly expressed mRNA, so the RNA content is here on the antibody side of this data structure. So you do see differences when you look into the proteome level. And finally, some cell surface proteins, CD3, CD19, and HLA. The HLA gene has a high allelic diversity, and hence the 10x cocktail does not provide any mRNA flow for that gene, but you can see it on the protein. Now, I'd like to introduce our products and services. So we currently offer one cocktail, a pixel human immune profiling antibody cocktail. It does comprise 53 targets and 5 isotype controls. The cocktail is lyophilized. It comes in single reaction tubes, and there are 4 tubes in a kit. So 4 single-use. They stain each up to 1 million cells. The cost is in the US $2,800, what it is. In Taiwan, you need to ask Genomics. It can be customized. So we launched the kit in November, and since then we have added about 100 antibodies that can be purchased as single antibodies. They are conjugated with oligos in that 5 sequence format. As I mentioned, developed exclusively for the 10x genomic single cell gene product. We work only with fixed cells, and you can find all these information online. This is the content of the cocktail. Secreted proteins, cytosolic proteins, nuclear proteins, material proteins. Those are the isotype controls. Don't go into any detail. The data is available online. If you have particular interest in certain antibodies, we can quickly look it up, but we have already that antibody so that we can spike it into the cocktail, or we also offer single antibody combinations. If this is something you want to do, but not matching your antibody requirement, you need a different cocktail. We can do custom cocktail. And in principle, I like to stress that in principle, we can do any antibody, any oligo, any antibody, any oligo, but of course, it should be for the 5-plex format in order to do quality control on those products. So this comprises Proteintech antibodies, those that we have already conjugated, or those that are in our catalog, that we can conjugate from other suppliers, or if you are using your own proprietary antibodies, we can conjugate those as well. We just need half a milligram of antibody. Then we have in the cocktail use two format, and we also provide custom validations, standard and standard without control. So if you have an antibody that you are interested in using, you don't know whether this antibody may work or may not work, we have an antibody labeling kit that labels as little as half a microgram of antibody. Very, very easy to step reaction, 5-minute sheets. We would encourage you to test your antibody with more flexible antibody labeling kit and check it in a flow cytometer, whether it can stay in the cell that you are desiring, and whether you get the results that you are looking for. The most critical step of the selection of an antibody for site-sec applications is the specificity of it. That's very important. So, custom validation website. There are two validation services, the standard one and the custom one. The standard one includes all of our projects. The cocktail is screened against human PBMCs or mouse sphenocytes. We obviously can't hold all of these samples you're working with. So if you have a differentiation or a special simulation or whatever, these are optional, require additional fee, but then fee depends on the work that is required, and whether we can get the cells or you need to ship the cells or whatever can be cost. Now, how does the workflow for the custom cocktails look like? So the cocktail needs to be finalized, and that will be done in a discussion with our lab at Diego Genomics. We will issue a code. We will sign the order. We will wait till all of the antibodies from other suppliers have arrived in San Diego, and then we will start the service. And up to 2 months later, when we receive all of the antibodies, we will conjugate the antibodies. We will do cocktail testing and optimization. We will have joint data review meetings, and then the final cocktail will be created and delivered to you. So that's a very open process. We share a lot of data. We have full control of the process, and I think here on the cocktail content, that's most important. So you will have certain antibodies for targets that must work. So we will put a lot of effort into your priority number one antibodies, but as a commercial company, if you select 400 targets and have 400 antibodies, we can't do that on all of those targets. So what are the deliverables? So single reaction tubes, individually labeled and packaged, with powerful formulation each. Any excess third-party antibody that is left over after the oligo conjugation will also provide it to you. And then you can get mixed cocktails with the intracellular and extracellular antibody mix or something. as you like. Everything is possible. The data report, that's the PDF, contains the UMAP for each antibody and corresponding RNA expression, the count matrices, and support a 700 guidance troubleshooting and help with data analysis if needed. So this is the prices of our customers up to about 200 antibodies. It is a number of reaction, a number of runs if you like. So if you want to stain 10 samples of 1 million up to 1 million each, it will cost you $10,000. If you order more, discount scheme will apply. If you require us to conjugate other additional antibodies that we haven't conjugated yet, the conjugation will also be charged. And our single antibody conjugation is at a price of $5. That is 10 micrograms of at least 100 runs. And we have those listed on our website. Also, this is UMAP, and we also have a PDF that I'm sure that we can share with you all the antibodies that we have. Also, the enhanced blocking reagent that's sufficient to block 4 reactions. So that's my presentation, my introduction into intracellular products from Proteintech for ABreal. I'm ready to take any questions. Thank you for your attention.